I'm Dr. Clifford Librach, and I'm a professor at the University of Toronto, and I'm also the founder and director of the Create Fertility Centre in Toronto, and I'm going to talk to you today about NIPGT-A, which is non-invasive preimplantation genetic testing using a combination of the embryo culture media combined with blastocoel fluid. So I have no conflicts of interest to disclose today. So PGT-A has really come a long way over the years, from starting in 1995 when there was FISH, and then multiplex qPCR, then to array CGH, and now to NGS, which has now become the standard. And NGS really gives us a much higher resolution for detection over 10 megabytes for doing whole chromosome detection for aneuploidies, as well as mosaicism. Now can detect over 30% mosaicism. So we've come a long way, and I think there is now the future, and that's what I'm going to talk about today. So for PGT-A, it's been shown very well now that aneuploidy increases dramatically with age, and so the number of embryos you have that are normal as you get older are less, and unfortunately the number of embryos you make as you get older are also less. So that's a sort of double whammy that people have as they get older, but it's something that we need to take into account when we do PGT-A. Now the other new finding is that mosaicism with NGS, we can now detect it. It doesn't seem to vary with age. There's similar rates of mosaicism as you get older. It runs in the sort of 10 to 15% range. And this is something that I think we have to do a lot more studies on to really understand the significance of mosaicism. So there are many advantages that people have come up with in terms of research on PGT-A, although it is still somewhat controversial. It basically overcomes the issue that traditional static morphologic assessment is really not enough. You look at an embryo at one moment in time, you're really not going to tell, you can't tell a lot about what happened before and what after, and also it doesn't really distinguish those embryos that are very well that are aneuploid. So we need more, and so the PGT-A has really reduced the maternal age effect, and if you see this graph, you can see that if you have a euploid embryo, the implantation rate seems to be the same at each age group, although you have less embryos, but you can really overcome the age effect, and that's very important. As well, it increases implantation rate, and that's something that's been shown in this meta-analysis here. It also decreases miscarriage rates, and that's something that's very important. We want to reduce that burden to the patient. It also reduces time to live birth. Now, if you look at cumulative rates, of course they're going to be the same, because eventually you're going to get to that embryo that's normal if you keep transferring embryos. So, but people, don't want to have to wait till they've had 5 or 6 transfers till they get to a live birth. So, reducing time to live birth is really important to patients. It also reduces the birth of abnormalities like trisomies, and reduces the burden of having to go through elective termination when there's an abnormality. And again, by using ESAT, elective single embryo transfer, you can reduce the multiple birth rate by just transferring one euploid embryo, which I think now has really become the standard for safety and many other reasons in IVF. So multiple birth rates are decreased as according to this meta-analysis here, and many studies have shown that. But there are pros and cons. I mean, we've come a long way, but yes, it does improve clinical outcomes, as I just said. It does optimize patient management, and it does give you comprehensive genetic information. But what are the limitations? It's invasive, so you take a biopsy, and I'll talk about that in a second. You're also sampling the embryo. You're taking, 4 or 5 cells out of an embryo to represent the entire embryo. It's like you poll for an election. You ask a few hundred people who you're going to vote for, and that's supposed to tell you who's going to win the election. It didn't work in the last U.S. election, as many of you know, but it doesn't tell you the whole story. Also, it's technically challenging because you have to train embryologists to do biopsies and do them well, and not everybody's as skilled at doing them and it takes a lot of experience. So it's been shown well that if you take too many cells from the embryo, you are going to reduce the success rate, and so the more cells you take, the more likely you're going to damage the embryo and reduce pregnancy outcome. And this was very well shown when we were doing cleavage stage embryos, and you're taking a large cell out of 8 cells, that it actually reduced the pregnancy rate, and we no longer do that anymore in almost all places around the world. Also, the other limitation is the sampling bias, as I mentioned. If you're taking an embryo that has a mosaicism and you select an area there's abnormal cells, you're going to call that embryo abnormal. And if you select an area that has a combination, that'll be mosaic, and so on. So there's always that risk of sampling bias in NIPGT-A. And again, technical skills are required. So what about non-invasive? That's what I'm really here to talk to you about, this new sort of technology that's come about. And just this is a little bit of the history of non-invasive. In 2013, a study came out showing that in blastocoel fluid there was DNA. And a lot of people didn't know that until that time. And then also in 2016, a study came out showing that you could take embryo culture media that's been conditioned by the embryo over a period of time, and you can actually get cell-free DNA from that. But we took it one step further, because we said, look, if you're going to be doing that, you may not get enough DNA. This may not be enough DNA. If you combine the two together, that we hope would be more, give us more of an ability to get results in a higher number of cases, and that's really what we've shown in our paper. So this is a sort of list of the papers that have been published in the last few years on this. And our paper, by the way, just the WGA rate, the whole genome amplification rate, is quite variable, but in our paper we had 100% using the combination. And I think that really made us sort of a pioneer in this, because it's really improved the detection rate. Also, we had a concordance rate of almost 90% versus trophectoderm biopsy. When we used frozen embryos that had been donated for research, we thawed them out, grew them for another 24 hours, and then tested it versus trophectoderm biopsy, and 96% if we took the whole blastocyst. And this gave us a sort of clue that maybe the non-invasive DNA that you're getting from an embryo that's been cultured may actually be more representative of the embryo, because it's coming from all of the cells from the embryo, and that may be a very important distinguish, and we may actually be more accurate when we do non-invasive versus a trophectoderm biopsy. And we did do an initial pilot on looking at 19 cases of live embryos that we tested, and I'll show you how we did that. We had 100% concordance, but it was a small number, and I'm going to show you some increased numbers from that that we've done more recently. So what are the pros and cons of this technology? Well, the pros, it is non-invasive, so you don't have to do the biopsy. It's reduced cost, because there's cost of doing the biopsy and handling the tissue and so on. And also it's less technically challenging. You're just taking fluids and you're analyzing them. The challenges are that there's a risk of contamination. You could have cells that are around the embryo that are humus cells that could contaminate from maternal DNA, and that's a risk of doing this procedure. And we've worked out ways to reduce that, but it may still be a theoretical risk. Also, we don't really know the origin of the cell-free DNA. Where does it come from in the embryo? How is it produced? And that's something that I think was very surprising to a lot of people, that we were able to do this in the first place, because DNA is supposed to be in the nucleus. Where is this DNA coming from? And that's also a question that, is it representative of the embryonic genome? One more thing is that it is require more sensitive genomic techniques to do it, and that's what we've been working out and troubleshooting over the last few years. So this is our workflow. We basically remove the corona radiata cells, we culture them for the first 3 days, and then on the 4th day we wash out the media and we do zona breaching, and then we actually transfer them to a fresh drop of media. And this is important because a lot of the DNA we found in the first 3 days gets degraded over time. So using that DNA, our results were not as accurate. So this really improved the results, and one of the troubleshooting techniques we developed in order to get the wonderful results that we were able to achieve so far. So this media then grows for another day to two until the embryo actually reaches the blastocyst stage, and then we collect it. And this is just showing if we would do the standard technique, we would collapse the blastocyst for freezing, transfer it to a biopsy dish, biopsy it, and then freeze it. However, if we do non-invasive, we can collect some media, and then we can test the media. And we use a collection of blank media as well as a control that's been cultured over that period of time. So this is the actual testing of the DNA, and so either the non-invasive trovectoderm, it's really a very similar workflow with Sureplex WGA as well as VeriSeq PGS, and we're able to then do the testing in a similar way that we do to the trophectoderm biopsy, but with some small modifications that we're working on. And so this just shows from our first paper in 2018 that you can see here is the non-invasive test. This is a monosomy 16 case, and we got exactly the same result with trophectoderm and whole blastocysts that were donated for research. So we could confirm that it does seem to represent the entire embryo as well. The question of where the embryonic DNA comes from is very interesting, and we don't really know. A lot of people feel that it may be apoptosis, that the cells are dying and they're releasing DNA. But if we theoretically think that's the most important or the likeliest source, well, you'd expect small fragments of DNA because of the apoptosis down to about 180 to 200 base pairs. So that's something that we wanted to test. Also, necrosis could also be but there'd be random degradation in multiple, and fragments would be different sizes. Also, extra chromosomal microDNA could be a source as well that's being produced outside of the chromosomes. Also, there could be blastomer fragmentation that could release DNA, and also fragment cumulus cells as well, and that would be the contamination by maternal DNA. And also the possibility of secretion of DNA, but you'd expect that those would be very, very small microDNA, small pieces of DNA that can be packaged into microvesicles. So these are all the sort of theories, and apoptosis seemed to be the one that's the biggest, the most likely one by many groups, so that's why we wanted to test that. So our hypothesis was that if it is a major source of cell-free DNA, we'd expect higher amounts of DNA in the extracellular fluid and the blastocyst fluid when you have embryos that have more apoptosis, that they have a lower morphologic grade. And we'd also expect smaller fragment sizes. So we tested this. So we know that embryos are graded in different ways, but we used this modified SARC grading, and we looked at embryos that were either higher grade or lower grade. We divided them up, and we had a total of 102 embryos that we tested. This is new data that's being evaluated for publication right now, and basically we looked at the good quality and the average quality, lower quality embryos as well, and we were able to determine that actually we could get amplification exactly the same regardless of the grading of the embryo, which was interesting and very important, because grading, if you have poorly graded embryos, you want to be able to get a result on those embryos because they can still be euploid. But the average size of the fragments was actually very similar to trophectoderm, and that was very important because that means that it's very unlikely this is due to apoptosis, and that's a very important finding that we'll be publishing soon. Also, we were able to get informative results from the NGS result in about 90% of the time, but a little bit less than trophectoderm, and that's why I think refinement of the technique still is required to get up to as high as we can to the trophectoderm biopsy results. So we're still working on that, and hopefully we can improve. And we have some newer techniques that we're using now that we haven't published yet that we think are virtually at that goal now. So what about the accuracy? Well, that's very important. You need to have an accurate result. We did look at that in our donated embryos and our early live embryos, but we have more numbers here. And was there a difference in accuracy between lower or higher grade embryos? And we found that the total versus trifectoderm accuracy level was about 98%, so virtually the same. We also were able to determine the gender in every single case, and we also were able to pick out the euploid embryos in 98%. There's even only one embryo out of 64 that did not correspond as being euploid in that whole group. As well, for aneuploid samples, we were able to detect almost all of them as being aneuploid. The ones that show as not the same, they just weren't concordant. They both showed aneuploidy, but they just weren't concordant which chromosomes were involved. And this shows an example where we could look at an embryo that has multiple aneuploidy in it with plus 10, minus 11, minus 20, and we were able to fully concordant results between NIPGT and trophectoderm biopsy. So this is really exciting, and we showed this is a normal sample that were fully concordant as well, and this is a case of a trisomy 16 that was concordant as well. We did have not many yet mosaic cases. This was one, and it showed that it was the same chromosome that was mosaic. It was a segmental mosaic, but it was it showed one had a gain, one had a loss. We're not sure how to explain that yet, but that it did show the same chromosome as being abnormal in that particular case. But we have to do more cases of mosaicism before we can know how accurate this technique is for mosaicism. And then so one of the questions also was that if we're doing the trophectoderm biopsy currently, can we use this non-invasive technique to use as a backup? In other words, what if you get no results with or inconclusive results with your regular PGT-A? Could you use this as a backup? And very interestingly, there's two cases I want to show you. This one was question mark minus 9, 30% mosaic, and when we looked at it with NIPGT-A, it actually looked at like a normal XY embryo. We were able to discern whether it was really abnormal or not, and I think that was very helpful for that particular embryo. Here's an example of a chaotic DNA signal. Anyone who does PGT-A knows that sometimes you get a signal that you just can't interpret like this after amplification, and this showed us that this was a trisomy 22 embryo, and we could get informative results in this particular case. So very, very interesting as a backup source for trophectoderm biopsy. So in conclusion, for this short presentation, blastocyst morphology doesn't significantly increase the amplification rates and concentration of WGA for non-invasive samples. And so there really doesn't seem to be an impact, and that's very reassuring that we can look at almost any embryo and tell what the result will be with this new technique. Also, the fragment sizes appear to be not what we'd expect for apoptotic cells, so it appears that the source of DNA is different than apoptotic cells, and we're in the middle of studying that to learn more about where this DNA is actually coming from. Using NGS and standard VeriSeq kit, we were able to show full concordance for sex chromosomes, ploidy, and whole chromosome copy number between the samples when we use the combination of the embryo media plus blastocyst fluid and the trophectoderm biopsy samples. And our findings provide evidence that this, combined with blastocyst fluid, has great potential for using it as a standard technique for testing embryos to avoid biopsy in the future. I think that it requires more study and more refinement still, but we're very, very close. And I think some of the newer studies that we're publishing and other people will really get us to a point where I think we can rely on this technique for most of our patients. So how could we translate it to the clinic? We have to do randomized controlled trials. We have to develop and improve on the culture, WGA and library and sequencing loads, and improve data analysis to get to that level that we can do with trophectoderm biopsy. We have to establish workflows for sample collection, analysis, and reporting. And I think this is a real analogy of this technique to where we were before with non-invasive prenatal diagnosis. that in the early stages the accuracy was low, but I think they've improved their techniques tremendously, so it's now used in the clinic on a routine basis, and I think that's where where non-invasive technology will go in the next few years. So I want to thank all the amazing people in our lab do this work, Dr. Majinkova and Valerie as well, and Ran, and all of the people in our lab that do this amazing work, and all of our people that do the biopsies that are amazing technically at doing the biopsies. Thank you very much, guys.